Alrighty, so let's take some time and organize the lymphatic system with this video. I have two maps that we're going to do. The first one looking at some of the structures. So how do we organize the main structures of your lymphatic system? All right, so with your two structures, we're going to classify them by being either a primary structure or a secondary structure. So to be a primary structure, think of that more like the school. It's where the cells are getting educated. So this is where they're born and where they're mature. So they're made here, or they mature there. So for these two primary structures, you're basically looking at the red bone marrow, which is going to be primarily for your B cell education. So this is B cell school. So think of it like that bone marrow right there is telling you that it's going to be for the B cells. And then for your thymus, that's going to be for your, yep, you guessed it, T cells. So thymus is going to be where the T cells are educated. So think of it as T cell school. Now, if we're looking at secondary structures, this is more like their work. So here's where they're getting their education. Over here is where they're going to do their job. So the job of the immune system, finding pathogens and attacking them. So secondary is where you're going to have these lymphocytes actually function. So under your secondary, you can have them separated basically if they have a capsule or not. So if they're fancily wrapped up with some dense, irregular connective tissue to hold them together. So if you don't have the capsule, you would be a nodule. And nodules are things like your tonsils or your mucosal associated lymphatic tissue, or MALT. So mucosa associated, meaning that you're in the mucosa. So what is the mucosa? Well, when you're looking at a common place for pathogens to come in, that could be along the digestive tract. The inner lining of that digestive tract, if you can imagine, secretes a lot of mucus so food can move through it smoothly. So why not have a bunch of these protective cells right there in that first layer that's making the mucus? That's your mucosa. So the inner lining of the digestive tract, the mucosa, has these areas where you have lymphatic tissue. So that's where the LT comes from. So mucosa associated lymphatic tissue, meaning that you're going to have like clusters of them. They're not anchored into place. They don't have a capsule kind of locking them into that spot, but they're grouped in an area along that digestive tract. That's the same thing with the tonsils. The difference is just location. So the MALTs are more small intestines, large intestines. So further down the tract, the tonsils are going to be up here more in the throat or pharynx area. Some of the tonsils that you will maybe be familiar with are the palatine ones. So it's going to be up towards the top and back of the mouth. The lingual ones, so that one's going to be behind the tongue, so lingual, think tongue. And then there's the pharyngeal one. And that's the one that's in the top of the pharynx, and the pharynx is the top of the throat. Under your secondary structures, these are the ones that have a capsule. They're formalized organs. And so with the organs, you're going to have the spleen, but that's where you will also have the nodes. The difference being what they're checking. So spleen checks over blood, so you'll have blood supply going straight to the spleen. All of these white blood cells are sitting there. You have reticular connective tissue there to catch any pathogens. Remember, those are the ones that look like thorn bushes. And then the pathogens will get trapped into that connective tissue, and all of these white blood cells and these lymphocytes that are right there can start the immune response. But they're specifically looking over stuff that's flowing through blood. So in the nodes, you also have that reticular connective tissue that'll look like a little trap. The pathogen gets stuck, and the lymphocytes will get activated. The macrophages will eat things. You start your immune response. The difference is the fluid. It's catching things in lymph. Remember, those are your two fluid connective tissues that's traveling throughout your body. And so with the lymph ones, you're going to see these nodes kind of being like, I think of them like the traffic stops, like where there's checkpoints randomly and they have a breathalyzer station and like the officers are right there. Same idea. You're going to have these checkpoints along the flow route of lymph. You have them in common places like axillary or elbow. You have some up here in the neck. You have some in the groin, behind the knee. So you're going to have them in major kind of like as you're moving from one region to another. So those are your structures for your lymphatic system, and then we'll come back and we'll talk about the cells. Okay, so this time now we're going to work into the more microscopic structures of our lymphatic system, looking at the cells, the workers that are actually doing the job of the system. So when we're looking at our cells, we're going to have three major categories. And so with these lymphatic cells, you have some that are going to just be wandering around and checking things out, making sure everything's supposed to be there. So they're actually reading the antigens. Remember, all the cells in your body have antigens on their surface. So they're checking them over to see if that antigen is, one, for your body, but two, if it's in the right spot. And three, they're also checking to make sure that that cell is healthy. So they can actually recognize something called a tumor-specific antigen. So what they're doing is basically surveillance. They're just checking things over. So the division here that's doing immunological surveillance, those are going to be your NK cells. NK cells, because they're called natural killer cells. So if they recognize something and they're like, oh wait, this isn't supposed to be here, or it's infected and it's not doing its job correctly, we need to get rid of it. So they're actually going to directly destroy a cell. So let's say we have a cell in the body that is sick. There's our little sick cell. We need to kill that off. So what we're going to do is we're going to release some toxins. The toxins that we're going to secrete are perforins. They're going to put pores in, so perforin, put pores in, and granzymes. Both are going to cause cell death by putting pores in or dysregulating the internal environment. The other categories that you're going to have are going to be more specific. So they actually care what they're killing, and they're also going to make more defenses for that pathogen. So over here, this is actually part of our what we consider our non-specific immunity. So that's non-specific on this side. But the next two that we're getting ready to go into, those are going to be specific. So they're actually mediating an immune response. They're going to not only directly respond, they're also going to trigger a lot of other things to get activated to go and hunt down the antigen that they have discovered. So over here, we're going to be in specific immunity. We'll have two major cells for this. So we'll have our T cells. And we'll have our B cells. With our B cells, we're doing antibody-mediated. And so with that antibody-mediated immunity, basically the idea here is that these cells, when they run across a pathogen, or if they're notified by something who did find the pathogen, they're going to divide. And when they divide, you're going to make two types. You're going to make a plasma cell, which is basically an antibody factory. So their job is to secrete a whole lot of antibodies. And so remember, antibodies are the ones that lock up to the thing on the surface of a pathogen. So let's say this is our pathogen here. And on this surface, we have a specific antigen. This antibody can lock onto it and create a whole variety of immune responses. So we are using the plasma cells as their little factories. The other thing that you make is a memory cell. This allows you to remember the shape of antibodies that you need for that specific pathogen, so you can't get sick with it again. This is what allows you to basically not get sick with the same exact strain of the flu ever again, or the same chickenpox virus, for example, is that your body is going to have these memory cells, so if they ever do encounter that particular antigen again, it can shift into becoming a factory very quickly. Now, if we go back to our T cells are going to do what we call cell-mediated immunity. Now, with cell-mediated immunity, it's called that because the cells are the ones that are actually in charge of the immune response. And there's 3 different types of T cells that are going to do this job. So it kind of makes sense that it's cell-mediated. You would have a lot of variety of workers. So we'll have our cytotoxic cell. You will have your helper cell. And you'll have a suppressor cell. So with a name like cytotoxic T, yeah, it probably kills things, right? Because it releases cytotoxins. It's going to release the same 2 that we got from an NK cell, but it also has a bonus one, an extra. So when you're looking at your cytotoxic T cells, again, you're going to have a pathogen. So let's say we have a bacteria that has come in. So we'll make him look evil. So here's our bacteria that's come in. We want to kill it. So we're going to release some toxins here. So we're going to release perforins to put pores in. We'll release granzymes. And we'll release something called lymphotoxin. So all three are going to cause cell death, again, by having a variety of functions and ways that they can do that. A helper T is something that is going to call for help, not surprisingly. So what they're actually going to release is going to be chemical messengers. The way that you're going to know that a pathogen has come into the body is because you're going to have these helper cells be notified by a healthy cell that has found a pathogen. So let's say we have a macrophage. This macrophage has done its job correctly, he's found something, and he just wants to show that off. So on his surface, he presents the antigens. It's actually called an antigen-presenting cell. So antigen-presenting cell is going to show it off to that helper T cell. As soon as the helper T cell sees that this phagocyte has found something, it's digested it, it says, ooh, there's probably more, can you go, coordinate a response to that? So it'll show off that antigen to the helper, and the helper is going to call in other things. So these chemical messengers are going to cause activation of your nonspecific defense, activation of other wings of your specific immune response. But the basic idea here is don't kill the messenger. He's just letting you know that he found something. So he's showing it to a helper T cell. So over here that's different because you have the infected cell or a foreign cell that the cytotoxic T is interacting with and directly killing it. So there's different cells that are communicating to the helper or the cytotoxic, and then you get different things released based on what you need. So healthy cell showing it off, release chemical messages to speed up or trigger the immune response. So these chemical messengers are going to call for help. That's different than the chemical message that will come from a suppressor T. A suppressor T is what you're going to use at the end of an immune response. It's your body saying, hey, we've done our job, let's ease off. And so this one is going to send chemical messengers, but now it's just like easy now, we're good. Like we've handled it, so we're releasing chemical messengers for both. The difference is the helper one, call for help, and the suppressor one, let's pull it back. We've done our job. We don't need to keep inflammation occurring, for example, or fever, and so we can ease off that response. So these are the major players for your lymphatic system. So remember, over here, these two are dealing with your specific immunity, over here, non-specific. The difference being here, kill anything that's not supposed to be there or it's not functioning, versus here, you're coordinating other cells or you're activating antibodies that go to a specific target. They know what they're actually hunting, and that's what they're going to go looking for. All right.